Okay, well, welcome to today's class. Like you know, we are still on Unit 7 of the Public Health Microbiology. So we are discussing about the mycology as a whole, which is the study of fungi. Like in the last class, we actually looked at actinomycetes. We actually discussed everything on it. So please, if you have not watched our last recording, endeavor to do that because it's actually going to help you. It's very simple, like is broken down into simple pieces that even someone that is not that doesn't have a microbiology background can easily understand. So, okay, so you have to go back and watch it in order to catch up. So today we are going over to Unit 2 of the Module 7. Okay? And the topic is on yeast. And under yeast, we have different genera we're going to discuss about. So basically, you have the Candida and then the Cryptococcus species. Okay, so we're going to be discussing about them, what the actually how they actually affect humans, and then the diagnosis and treatment of each and every one of them. Okay, so let's get started right into it. So, um, what are actually yeast? I know we've heard a lot about yeast, and we want to maybe just have a one-sided view about yeast. Majorly, um, most people know yeast as maybe what we use to rise bread, like put inside bread, a rising agent, but there's more to yeast than that, okay? So yeast are simple celled organisms that actually reproduce mainly by budding. Okay, so they are unicellular fungi that reproduce by budding. I'm going to explain what budding means with a pictorial example. So they are found majorly on the skin, on the mucous membrane, in the soil, and then in the environment generally. So they can actually be a normal flora in all these especially in the human body, or they can be pathogenic. Okay, why many are harmless, there is normal flora, like I said, like the Saccharomyces cerevisiae, which is called the baker's yeast, and is majorly used as a rising agent for baking of bread and for other um, food. Okay, some can cause opportunistic infections, especially in people with weakened immune system. In as much as yeast are beneficial for humans, some are still a no-go area that can cause diseases and infections. Okay, so when these are grown on culture media, when maybe doing diagnosis, the colonies are usually visible on the plate in 24 to 48 hours. And one of the general characteristics of them is that they are soft, they are moist, they have a moisty colonies resembling bacterial cultures rather than mold. Okay, these are just the general characteristics of yeast. That to give you a general overview of what yeast is all about. Then there are three major human pathogenic yeasts that we're going to be looking at tonight, especially according to our material. There are over like there are a lot of them, but for the sake of uh, um, our topic today, and also according to our material, we're going to be concentrating on three main pathogenic yeasts. Number one is the Candida albicans from the Candida species, and then the Cryptococcus neoformans and the Cryptococcus gattii from the Cryptococcus genera. Okay, so these are the three major ones we're going to be looking at tonight. And if you're able to cover these three major species, I bet you any question they ask on yeast, you are covered. Because one thing is very special about the now exam, that is our school, is that we they will never set question out of what is in the material. It's very, very pronounced. You can never set question out of what is in our material. I've not even done introduction. I think that's my bad. For the sake of new people that are joining us, I haven't really introduced myself. So let me just do it briefly. My name is Jane Nena Ezekai, and I am a public student of now National Open University, and I am currently in third semester. So for microbiology, I'm done writing it, and by the special grace of God, I passed the course. So I'm trying to teach my dear colleagues So I'm going to write a course in coming weeks on on the things I know. Okay, so I'm just trying to impact or give back to you guys. So on a disclaimer, I'm not a lecturer or parading as one, but I hope to be a lecturer soon because I love teaching. So that's it about me. So let's go over to continue from where we stopped. So I'm just trying to explain that if you're able to get everything I'm going to explain on this today, right? I'm very sure and hopeful that you're covered on any question that is going to ask on this particular topic. Okay? So let's discuss on the Candida species. So Candida is a gram-positive polymorphic yeast that measures about 10 to 12 microns in diameter. Okay? Polymorphic means is that it can exist in different forms. Number one, it can exist as the yeast cells, it can exist as a hyphae or pseudohyphae. Okay, so this ability to assume various forms may be related to the pathogenicity of this organism. So the ability to change to different forms, maybe in different environments, can be why they are able to penetrate the host and cause infections. Okay, so it Candida can grow overnight from bacteria and fungal media, and also produces what is called germ tubes. I'm also going to show you a picture of germ tubes and what it actually does, okay? Then there are more than 20 species of the genus Candida that cause diseases, and they all causes a disease infection called candidiasis. At one, I think some point in our life, I think we must have heard of this candidiasis. Maybe as a woman, at one time in life, you must maybe go to hospital and they actually diagnosed with yeast infection, candidiasis, by fungi, and others. So I think this is a kind of familiar word to all of us. So the most common species that actually causes human infection is the Candida albicans. Like I said before, there are numerous other species, there are more than 20 species, but the one that majorly causes human infection is the Candida albicans, and that's one we're going to be concentrating on. Okay? So Candida albicans is an endogenous organism. Endogenous means that they are found inside the body. We have endogenous organisms and we have exogenous organisms. So endogenous are the ones found inside the body, and then exogenous are the ones found outside the body. Okay, please know the difference. Then it can be found in 40 to 80% of normal human beings. Yeah, so it's in this Candida albicans can be in normal flora in the human body, which means it doesn't cause diseases as a normal flora when it's found in the right environment. But whenever maybe the environment or the pH is distorted, it can cause disease. Let me take for example, like for example, it's present in the mouth, in the sputum, in the gut, that is in the stomach, and then in the vagina of a woman. Okay, let's take for example the vagina. You know, when the pH of the vagina is normal, this candida albicans can be found there as a normal flora living there just like that. Okay, but maybe a woman is using a lot of all these chemicals in the body and restore the pH of the genitals. This candida albicans can thrive and can cause overgrowth of this yeast and start causing infection. That's why you start some itching, discharge, abnormal discharge, and then smelly discharges. Okay, so they can be found just as a normal flora in the body. So it may be present as a cancer or a pathogenic, just what I explained. Cancer means they can just be found there, not harmful to the body. They're just there on their own, living there, maybe because of the comfortability and then the suitability of the environment they found themselves in. Or they can be pathogenic when the environment is distorted. They can tend to be harmful. Okay, so infections with Candida usually occur when a patient has some alteration in the cellular immunity. There's a normal flora or normal physiology when there's changes or alteration, just what I explained. And then patients with this decreased cellular immunity have decreased resistance to fungal infections. He's talking about those immunocompromised patients, maybe those living with HIV or diabetes. They have low immunity and they can come down with these infections very easily. I hope you are all following. Please, you can just chat on the drop a comment on the chat box if you are following this teaching, please. So like I explained about that yeast
actually reproduce asexually by budding. So what budding actually means that the new let's say the new or daughter cells actually are gotten from the parent yeast cell. From the picture from the A on my left hand side, you can see the nucleus, you can see the parent yeast cell. So when they tend to form balls, they're kind of breaking out from the parent yeast cell, and then um part of the nucleus also go with the newly formed daughter yeast. You can see the daughter yeast that have been formed. Okay. Then part of the nucleus they divide asexually. Okay. They don't need like any maybe external, just like sexual reproduction and asexual reproduction. So that's what budding is all about. The daughter's yeast is actually gotten from the parent's yeast just by breaking and then sharing of um its nucleus, okay, or the chromosomes, as the case may be. Then for the germ tubes, the germ tube is one of the major characteristics of and I'm going to mute everyone. So the germ tube is one of the major characteristics that is used in identifying Candida albicans in the lab. Okay, so they are outgrowths that develop from yeast cells within 2 to 4 hours of incubation in serum at 37 degrees Celsius. So they are virulence factor aiding in tissue penetration and immune evasion. So what they actually do, they help, they actually aid these Candida albicans in increasing the like pathogenicity or invasion of the host cell or the host body. Okay, so if you want to diagnose, which we are still going to explain in diagnosis of the Candida albicans, you're going to use the germ tube testing. Okay, this germ tube testing helps to identify or differentiate these Candida albicans from other Candida species because it's the only one that produces the germ tubes. You can see the spore of the Candida albicans, then the germ tubes actually growing just like a hypha or a filament. So whenever you put it or you grow it in a serum for 2 to 4 hours, you actually know if this person is battling with Candida albicans or yeast infection. I hope you understand. Okay, so let's go back to um, the types of candida infections. So there are three major types of candida infections in humans. We have the oropharyngeal candidiasis. This oropharyngeal candidiasis is majorly, it can also be called oral thrush and the candidiasis of the mouth. So it majorly affects the mouth area. Okay, so the major characteristic is that it can be seen as white patches on the mucosa of the mouth, including the tongue. Okay, the affected area can become inflamed and they may be difficult in swallowing, so it can cause pains due to the inflammation. The cracking and inflammation may also occur around the mouth, and this is called oral cheilitis. So when there's cracking and inflammation around the mouth area, then this infection can also spread to other parts of the body, and when it spreads to the esophagus in the gut, it's called esophagitis. Okay, then they are majorly found in immunocompromised individuals, that is the HIV patients or people that are just undergone organ transplants or use of corticosteroids drugs. So this candida infection can actually invade more on these people that are immunocompromised. So the CDC, which is Center for Disease and Control, estimate that between 5 to 7% of neonates develop oral candidiasis. Neonates are newborn, okay? They develop oral candidiasis. And I want to ask us, do you know why? Or can someone just try and answer why 5 to 7% of newborns develop this oral candidiasis? Can someone try? Like why? Do you think why do you think that newborn tend to develop this oral candidiasis?
I think the reason is that most of them put their hand in their mouth,
Okay.
and of the hand is contaminated due to people carrying them up and down and stuff like that. And another thing is from the mother's nipple region when they
Okay.
suckle the breast of the mother. If
Okay.
the nipple is properly washed before feeding the baby can develop oral candidiasis.
All right. That's beautiful. Thank you
Can
so
I
Uh,
ask,
much.
may I
something?
also add?
Yes, yes, you can. Sure.
Okay, I also
Okay.
think the level of their immunity also
Better, yeah.
predisposes them to the
Exactly.
infection, like
Yeah,
low immunity. Yeah.
that's
Can I
beautiful.
add?
Yes, please.
Or maybe kissing newborns since their immunity is very low. Maybe an
Mm,
adult who has it kisses them maybe on the cheeks, on the lips.
yeah, that's a good idea. Yeah, you're right. So all of us are correct, okay? Because newborn, they have a very
Exactly.
fragile immunity, okay? They are just um being given birth to, and before you know it, they are down with all these, maybe all these infections, because they their immunity has not been built up, okay? So they can come up with it. That's why up to 5 to 7% develop oral candidiasis, because the body cannot really fight these infections. Okay, so this is a pictorial example or a pictorial view of what oral thrush is. You can see the white patches in the mouth, even the tongue area affected. So this is what oral thrush actually look like in humans. Okay, so let's talk on the second type of candida infection. We have the genital or the vulvovaginal candidiasis. This is the mostly used in the hospital. Like whenever you go to a hospital, you must have heard that a woman has yeast infection or have yeast infection, especially those that work in the lab. Okay, so this mostly occurs in women, and it's estimated that most women will have at least one episode of vulvovaginal candidiasis, at least once, at least some point in your time in your life you must have had an episode, maybe because of toilet infections, use of chemicals in the genital area or other things. So, and it's very, very rare in men. Okay, so the symptoms include itching, a burning sensation. You can see the itching that you'll be very eager to itch your genital area, is like that itching that is very sweet. Mhm. So we also have a burning sensation, abnormal discharge. Then in men, the penis can have itching rash, but you know it's very rare in men. So women that are at more risk of the infection is those women that are pregnant, that are diabetic, that use broad spectrum antibiotics, that use corticosteroids. Corticosteroids are medicine that you use to treat like rheumatoid arthritis, inflammatory diseases, asthma, allergies, and all other diseases. Okay, so those women that actually make use of this drug for a very long time, or maybe the women that are pregnant, you know, if a lot of women when they're pregnant, the hormonal changes can actually affect the pH of the genital area, and they can come down with this yeast infection. Let me just say, from we did a practicum in IDP that was last week. So we carried out our practicum in IDP, and we did medical outreach. We have a focus on pregnant women in IDP. So we did a mini outreach for them. And from the personal consultation with the pregnant women, almost all of them are battling with infection. Like it was just so like overwhelming. Almost all of them they came to complain, having itching, having this discharge, having this one. Ah, it's paining me. You know, most of them are not educated, so is to just be explaining it with their whole heart. You just notice like it's a kind of recurrence of especially in pregnant women. Okay. So we have the third one, which is invasive candidiasis, and also be called candidemia. So this occurs when candida, which is normally on the skin or the gastrointestinal tract, enters the bloodstream where it can disseminate to other organs. Okay, invasive means that something actually an intruder. It leaves where it's supposed to be and enter into other organs. Okay, there are about 46,000 cases of candidemia each year in the United States. This according to research. Okay, and they are majorly nosocomial infections of people who have central venous catheter, who are immunosuppressed, who take broad spectrum antibiotics, and also who show neutropenia. Neutropenia are those who have low neutrophils. Neutrophils are white blood cells which helps to fight diseases. So those people that have that suffer from neutropenia, that is with low neutrophils, actually can actually come down with these infections. So do you know what's nosocomial infection? What are nosocomial infection? Who can try? What are nosocomial infections? We say that candidiasis are often nosocomial infections. Do you know what is nosocomial infections? Who can try?
Yes, they are hospital-acquired infections.
Exactly, they are hospital-acquired infection. Maybe you went to a hospital to treat malaria, and before you know it, you've gotten cough or something. Just any disease you get from hospital, apart from the one that you actually go there to treat, is called nosocomial infections. Okay, so the symptoms of these candidemia are fever and chills. And most times when you have fever and chills that do not respond to antibacterial agents, maybe you use antibiotics to treat fever and chills, and most times it's not responding. So you can just go and test for invasive candidemia if it's there. Okay, so let's talk on the diagnosis of Candida species. The clinical material, that's the things that is going to be used for diagnosis. You can use the blood cultures, you can use vaginal discharge, especially for those that have the yeast infection. You can use urine, feces, nail clippings for subcutaneous candidiasis, or material from the cutaneous or mucocutaneous lesions. Okay, so these are the things that they may you can actually use for um, investigating of this candida species, especially in the laboratory or in the hospital. So number one, you can do microscopic examination, okay, where you have to stain this organism. And most Candida albicans appear to be gram positive. That's those ones that appear purple. Hope you've read about your gram stain, because they may ask you what are the steps in gram staining technique. So they can also be oval shaped and may show budding yeast or pseudohyphae. I've explained what budding means. So when you stain them and observe them in a microscope, you can check for these characteristics. And if you see any of them, that means it's Candida albicans that is actually um, there, okay? So you can also culture them on culture media, like the Saboregastrus agar, which is majorly used for fungi. It encourages fungal growth. So when you culture the organism there, the colonies appear to be creamy, smooth, and off white to off-white color. That means you'll be suspecting um Candida species. So colonies are just growth. If you are used to laboratory, you notice you'll be conversant with all these things where you tend to use culture media, when you tend to grow different organisms on a culture media that is favorable for them. So when maybe you grow them in a suitable environment that is being required, and when you observe their growth, you'll be able to identify which organism is growing on this culture plate. They also have the jam tube test. If you listen from the beginning, I said the jam tube test is a very unique testing, especially for the Candida albicans. So when you actually the process is that the yeast is incubated in a serum. It can be a human serum or the sheep serum at 37 degrees Celsius for 2 to 3 hours. Okay. So after 2 to 3 hours, you have to observe the serum. If there is a jam tube formation under a microscope, it's confirmed that it's Candida albicans or Candida dubliensis, which is rare, but Candida albicans are the major ones that actually form this jam tube. Okay. Then we can also use polymerase chain reaction in the diagnosis, where you get to check the chromosomes. That is the DNA sequencing of these Candida species. Okay, for treatment, we basically use antifungal drugs like clotrimazole tablets or a nystatin suspension. We also use like fluconazole or itraconazole or amphotericin B. So most of these drugs you can use in treating them of all these candida infections. Okay, so if there's an artificial heart valve or an indwelling catheter becomes infected, like I said, it can occur in let me go over to this. They're often in nosocomial infections of people who have central venous catheter. So if catheter that is used for a patient is actually infected, that catheter must be replaced. Okay, it must be replaced so that it can actually get rid of this candida infection. So I've been explaining of Candida albicans throughout. It's not only Candida albicans that can actually cause disease. There are other Candida species, and they account for an increasing number of nosocomial infections. Okay, so these are other different Candida species. This is not found in the material, just like an extra knowledge I feel is going to be important for us. So we have Candida albicans. Just to let you know, there is no only Candida albicans. We also have Candida glabrata, tropicalis, Parasiloxis, Crocea, aureus, and dubilensis. Okay, and each and every one of them are have resistance concern. You'll be hearing of antimicrobial resistance. Okay, so where organism is um refusing or inability of an organism to actually be successful to a drug or react to a drug. Okay, they tend to be resistant because over time they tend to change or develop in such a way that the drug will not actually treat them. You see someone, maybe let's say for example malaria drug, you see you're taking maybe artemether, and over time when you take artemether, it's like it's not doing anything in your body. You see that that particular malaria organism is now resistant to that drug. You have to upgrade to a higher version. And antimicrobial resistance is actually a public health concern because a lot of people are abusing antibiotics, and it's really a concern because they're not really, the body is not reacting to any drug. People are coming down with diseases, and when you give them drugs, they are not, thing is not going, and they keep coming. So it's really, really a great concern. Okay, so that's it about different other species of the Candida general, and then the notable features and the resistance concern. So this for extra knowledge, okay? And it's not in the material, so you can skip that and store it in the other part of your brain. So at this point, before we go over to the Cryptococcus general, I just want to make a little appeal to each and every one of us. This class is a free will class. Like, I do it from the bottom of my heart. Yes, it's something I derive joy in doing if I can. Okay. So, and with the little time I have from my schedule, I tend to put all these things together for each and every one of us. And I break it down in a simple way that even though you're not in a microbiology background, or you don't have any background when it comes to microbiology, you'll be able to understand. Okay, at least when you understand, and if this question are asked in the exam, you're able to write something. The aim, as always, is to write something during the exam. We don't have to submit an extra empty sheet. Okay, so that's the aim. And I just want to make appeal because I'm using a lot of data to compile all this. Yes. So a lot of data goes in hand, and then subscriptions, you know how it can be crazy. So I'm making appeal and also a commitment from you guys, okay? Because we have like some weeks now to the exam, and if we're able to make these commitments, I promise I'm going to bring out time from my busy schedule to continue with this, okay? So the appeal is that we're going to make a commitment fee, and I'm going to tell you what this money is for. This money is not for you to pay me for my time, because I take a lot of time, and I'm not even sure how much I can tell you to pay me as of now. Even 100,000 will not be enough because I do a lot of research. I'm not, if you notice my slide, they are not basically what's in the material, but I try to make it to be specific on what is in the material, because now cannot ask you what is outside the material, but I try to break it down into simpler form that to be easily understood and straight to the point. If you notice the material, a lot of pictures, a lot of things that we don't even need. Okay, so this money you're going to commit is just basically for data and for subscriptions. Okay, and it's going to be something very small that you can easily pay, because I know how the economy is, and then the exam is a little nearer. So trying to get a lot of us out of the program out of this discussion forum is something that I don't want to. Okay, so the commitment fee is going to get you access to our discussion forum probably every night. Okay, or as I tend to make it. Then it's also going to get you access to the recordings, because I tend to record this class so that you can get to watch it over and over again till it sticks to your head. So if you're able to commit, you're going to get access to all these in a private group. Okay, so the commitment fee is just 5,000 naira, and it's going to be till you start your exam, as long as you want. Even after exam, you still have access to all this recording. Okay, so the 5,000 naira is basically for data, the data support, and then just for my own research and for my own time I put in this, it's free. It's something I love doing. If I have my way, you can. I don't want anybody to pay for it, but data is now is very, very high. So for your data support and then for your commitment, because the Bible says, you see, where your treasure is, that's where your heart lies. I know where people commit money, they'll be more serious. So I want you to be more serious. So the money is basically for data support and then for your commitment. Okay, and it's a minimum amount of 5,000 naira. So if you're able to pay, I'm going to be adding you to a private group so you can just have a private discussion for those that be able to commit. So this is just what I want to make an appeal for us, for those that are interested. See, please, it's not compulsory. It's just for those that are interested and those that want this discussion to continue till exams. Okay, so that's it. You can just commit to this account. So if you do it, you can send me a private message. My number is on the WhatsApp group. I forgot to add it here. You can screenshot this page. You can just send me a private message. So I will add you to the personalized private group for discussion forum. Like I said, it's not for everybody. And this one I'm doing now, this particular recording and this particular class is free. It's for everybody. For the subsequent ones, it's going to be for those that commit. Thank you so much. So let's continue. So the next one we're going to talk about is the Cryptococcus species. Okay, so cryptococcus are still yeasts, which are a group of fungal species that can cause serious infection called cryptococcosis, okay, which is a potentially life-threatening fungal infection. So in humans, there are basically two major species that actually cause human diseases. Number one is the Cryptococcus neoformans and the Cryptococcus gattii. So one thing, how they normally ask this kind of question is they can tell you to write on Cryptococcus gattii. Number one, you have to write like general characteristics about them, write their infection or mode of transmission, then write the diagnosis and treatment. If you're able to write this basic, you are good to go. You get your full mark, no cap, okay? So in each of them, just aim to have like at least 1 to 3 characteristics of each of them. Then the mode where be that like the mode of transmission, the how they are in the environment, okay? Then know their symptoms. Okay, if this affects humans, where do they really affect? How do they the symptom shows, and then the diagnosis and the treatment, they are very important in case if you are asked on any. The same thing with Candida albicans. I've explained all of those in the previous slides. Okay, then let's go to Cryptococcus neoformans. So just pay attention so I get to general characteristics and the symptoms, diagnosis, and treatment. So for Cryptococcus neoformans, they are very distinctive yeast. It's a round single cell yeast surrounded by capsules. Okay, so the cells, which are spherical and 3 to 7 microns in diameter, produce what we call buds, which I explained both, that characteristically are narrow base, and the organism is surrounded by a polysaccharide capsule. And what this capsule is, this capsule acts as a virulence factor for this organism when they want to invade, especially the human host. This capsule actually help them to maneuver a lot of human maybe the immunity trying to fight it. The capsule, they are protected in a capsule that actually aid them to invade their host, okay, and also produce an enzyme called phenol oxidase. So this phenol oxidase is also another virulent factor that helps it to maneuver and cause infections in humans, okay? The ecological niche, that's where they are majorly found in pigeon and chicken droppings. These droppings, like in pigeon shit and then in chicken droppings, they are majorly found there, okay? Then their transmission is through respiratory system. When this is actually inhaled through the lungs, okay, they can be transmitted. If you inhale the spores, they can actually go into the lungs and start causing infections. So they cause the disease called cryptococcal meningitis and then the lung infection, especially in immunocompromised. So these immunocompromised people, they're always liable to different infections. That is one notable characteristic you have to note for all these things. Every disease, every microorganism are majorly in immunocompromised persons. So if you don't even know what to write, just write the majorly cause disease in immunocompromised or people that are living with maybe HIV or diabetes. That's one expo. Okay, so the yeast may also form a mass in the mediastinum. So mediastinum is the area in the chest that contains the heart, major blood vessels, trachea, esophagus, thyroid gland, and lymph nodes. So this mass is just like a growth they form in this area and is called the cryptococcosis. Okay, so when they form this mass or these growths, when they're able to invade the host, that is the human host, and come to the chest area and start forming a kind of lumps or mass, it's called cryptococcosis. Okay, so the diagnosis for these Cryptococcus neoformans. So let's talk about the clinical material that you can send to the lab. We have the cerebrospinal fluid, which is the CSF cerebrospinal fluid. We have the biopsy material, especially when it affects the the area of the body is affected. You can cut out the tissues and go to the lab and investigate. You also have the urine, okay, but he said that the urine is very rare. Okay, it's a very rare material to be required in the lab. Then different method you can use in diagnosis is culture methods. Number one is that it can be grown overnight on bacteria or fungal media at 37 degrees Celsius. And then when it grows, it forms a creamy white mucoid because of the capsule colonies. Okay, so growth in the culture is usually visible within 24 to 48 hours. And then as the culture ages, it turns brown due to a melanin production by the phenol oxidase. I told you it produces phenol oxidase as a virulence factor. And then when you leave this culture media for about 48 hours or beyond, it tends to turn brown due to the melanin produced by the phenol oxidase. Then you know that this is a candida Cryptococcus neoformans that is actually growing here. Okay, we also have serology testing where you use the latest agglutination test, which can be used as a prognostic test to know if this patient is actually having this yeast infection. Okay, so as the patient improves, as the patient heals, the serum antigen titer will also decrease. This titer, I think, should be in module 6, yeah, for the all the serology testing. We are still going to discuss about them, the agglutination test, and a lot of the lots. Okay, so when you do the test, you get to know if it's basically on antigen detection. Okay, then we also have the staining technique, use of a special stain called musicanamine, which stains the capsule. Okay, so identify the organism in tissue sections. So what about treatment? Treatment is still using antifungal drugs like amphotericin B and then 5-fluorocytosine, which is 5-FC, is an oral drug. So you can use both these drugs together. You can recommend it to a patient that is battling with these Cryptococcus neoformans because the two of them are synergistic. That is, they work together. If you recommend like amphotericin B, it may not work as efficient as recommending the both of them. So it's always recommended that you recommend the two drugs at a time because they work synergistic. Okay, so that's it about the Cryptococcus neoformans. So the next one is the Cryptococcus gattii. So they are closely related to the neoformans, but there's a slight difference, okay? They are majorly found in the tropical and subtropical regions of the world, but has recently been found in the western United States and western Canada, unlike the neoformans. If you notice that the neoformans is found generally in the environment everywhere in the world, the neoformans can be found. But when it comes to gattii, it's one of the different between the two. They are majorly found in the tropical and subtropical areas like Africa. The cryptococcus they are there, okay? But in neoformans, they are found everywhere in the world. Okay, just note one of the difference. Then for transmission, you can get cryptococcus gattii from inhalation of fungal spores, which lodge in the lungs. So these spores, when it enters into your lungs, it transforms into a dividing yeast cells and it can disseminate to other parts of your body through the blood. Okay, and this can cause what we call meningoencephalitis or lung infections. Do you understand? So you can actually get them by inhalation through the pores. Majorly, this is found there are some specific trees that this Cryptococcus gattii can actually be found in. I'm going to check on them and tell you maybe on the group, okay? So when you actually inhale these fungal spores, it gets your lungs and it starts dividing or reproducing. And then when it disseminates into different parts of your body through the bloodstream, it can cause lung infections or meningoencephalitis, okay? So the appearance of symptoms usually occurs several months, like 6 to 7 months, because most of them tend to remain asymptomatic, especially someone that is healthy or have a very healthy immune system. You see that your immune system will actually suppress it. It's not give it that time to explode or disseminate to other parts. So it can stay in your human body for several months, even after breathing the spores, okay? The disease is not contagious, so it cannot be transmitted. So it's not contaminable. Okay, so the people at more risk are those that are immunocompromised, like I was saying. Whenever they ask of any of all these species, just say the people at most risk are those that are immunocompromised, that have low immune system, like AIDS and cancer patients, or people on immunosuppressive therapy. So they're always at risk of any of all these infections. Okay, so we have symptoms. Symptoms of these Cryptococcus gattii. Number one is malaise, like fever and headache. We have cough. We have shortness of breath. We have chest pain. We have neck pain. We have nausea, light sensitivity, confusion, especially when it's caused by these encephalitis, which affect the brain. So you can have like brain seizures and confusion. Okay. Then diagnosis. As usual, the material you can send to the lab is the biopsy material, the cerebrospinal fluid. Okay. So when culturing this organism, to distinguish it from the Cryptococcus neoformans, they can be grown on a media called canavine glycine bremotamol blue agar. Okay, so when you this is just a kind of a culture media used in growing microorganisms. Okay, so when you grow this organism here, only Cryptococcus gattii turns this media blue. Only Cryptococcus gattii turns it blue. So when it turns blue, you know that actually is actually Cryptococcus gattii and not neoformans. So neoformans cannot turn this culture media blue. Okay, it's only Cryptococcus gattii that can do that. So it's basically this cultural method that can use in distinguishing this Cryptococcus gattii from Cryptococcus neoformans, or you can do PCR, which is polymerase chain reaction again. Okay, so treatment is by antifungal agents for up to 6 months, depending on how invasive the infection or the disease is. Okay, you can use amphotericin B in combination with flucytosine. Okay, in milder cases, you can use fluconazole or itraconazole. So these are just antifungal agents. And if you notice, these drugs I've been mentioning, these antifungal drugs are recurrent, especially most of all these fungal infections. Okay, so whenever maybe you're mentioning treatment, just check out for those drugs that can be used for all of them, especially this fluconazole or itraconazole. So you can just write them when it's come to treatment instead of maybe cramming strange drugs. Just note those ones that are common. Okay, those similar things, because all these are fungal infections, so they must have something common, like the use of this drug. Some drugs you can use in treating almost all of them. Okay, so just note those drugs that you can actually remember in case if you are asked to talk on the symptoms or the treatments. Okay, so that's it about the Cryptococcus gattii and the Cryptococcus general. So the next topic we're going to be talking about is the dermatophytes. So another the dermatophyte, we have the Trichophyton species, Microsporum species, and then Epidermophyton. I think we're going to finish this in another section. Okay, so this is almost 9:30. So in another session, probably by tomorrow, we'll continue with the dermatophytes. I think that's fair enough. By tomorrow, we'll continue with the dermatophytes. But for now, we are rounding up with the yeast explanation. Anything on yeast, everything on yeast. I think we have done justice to that. This is all you need to know according to the material. Okay, so that's all for now. Then we'll continue again tomorrow on the dermatophytes. But this is going to be exclusive for those that make commitment. I'm sorry for that. Thank you so much for your attention and see you in next class. So if you have any question, you can
Thank you
ask
very
now.
much.
Thanks so much, sister.
So
God
much.
bless you.
Increase you.
Thank you. So do you have any question?
Thank you so much. Really appreciate.
Yes, I'll drop the recording as usual. That's for Coin. You're asking if I can drop the slides. For sure, I'll drop it as usual.
Thank you. Thank you very much.
Thank
Really
you.
appreciate it. Today the network is not friendly, but
So for those that
I
for
can
those
hear.
that
Okay. Hey,
the
can you hear
network is
me?
bad.
Hello. I can hear you now.
interested in paying, do they get access to like the
Oh my God. I'm struggling to hear you.
interested in registering?